Good afternoon, everyone. I extend a warm welcome to everyone present for the inaugural session of this newly launched series, Innovations in Medicine, Symposium for and by the students. It is indeed a moment of great honor and privilege as we gather here today for this special occasion. We are delighted to welcome a distinguished guest, an eminent scientist, and a thought leader in the realm of science and policy, Dr. Geetha Vani Rayasam, currently serving as the head at HRDG. She'll be virtually joining us for this session. This symposium, Innovations in Medicine, envisioned by the director at CSIR-CDRI, Dr. Radha Rangarajan, is being initiated as part of the Platinum Jubilee celebrations of the Institute and will continue to be held once every year. The symposium is designed to provide an engaging and educational platform for showcasing the journey of recently approved drugs by the US FDA. With this said, I would kindly request Director Ma'am to deliver the welcome address. Ma'am, please.
Good afternoon, faculty, and my dear students. I welcome you all to today's event, and a special welcome to those joining online and to the students from Lucknow area academic institutions, Amity University, Integral University, Lucknow University, Maharishi University, Hygieia Institute, Jeevika Institute of Pharmacy, and Sri Rajendra Singh College of Pharmacy. A warm welcome to our chief guest, Dr. Geetha Vani Rayasam, Head Human Resources Development Group, CSIR. She's a distinguished scientist, as Neha mentioned. She has a PhD in biochemistry from the Indian Institute of Science and has postdoctoral research experience from the Institute of Genetics and Molecular and Cellular Biology in France and at the National Institutes of Health in USA. She has a particular interest in drug discovery, particularly in the area of TB. Many students here know how much change she has brought about by streamlining many processes, including fellowship disbursements, greatly improving the quality of the PhD experience for our students. So, very warm welcome to you, Geetha. Thank you for joining us. We wish you had been here in person, but under the circumstances, we are happy that you can still join us online.
Thank you.
Now, our esteemed speakers for today are our own students, CDRI students. In fact, they are the organizers and presenters for today's symposium, Innovations in Medicine. As Neha said, we are celebrating our Platinum Jubilee 75th year, and we are commemorating the year with a series of events. In addition to the translational research and the Innovation in Health series, in which we invite speakers from outside to give talks, this symposium is special in that it is focused on CDRI students. Moreover, it's meant to be a unique learning experience, with our students becoming the teachers for the day. When I was in school, we had a very unusual kind of celebration for Teachers' Day. The school management decided that on September 5th, all the teachers should get a break from teaching. So instead, all the senior students became teachers, and they went into the classrooms and they taught as per the lesson plan. Very often, things would go just fine, but sometimes the student teachers would decide to not only teach as per the lesson plan, but also imitate the style and the mannerisms of the teacher they were posing as. And as you can imagine, this led to many comic scenes, lots of laughter, lots of chaos. But that's not the kind of celebration we are here for today. When I said our students are going to be teachers today, what I meant is that they will present a thorough, in-depth analysis of the science behind two breakthrough drugs that were approved by the FDA in 2024. They will present what they have learned and in the process create a learning experience for all of us. Now, why this particular type of symposium? Well, every year between 40 to 50 drugs are approved by the US FDA for human use. Each drug is backed by science and innovation. The drug may have started off in academia and wound its way through startups and large companies, or it may have entirely stayed in large companies, or it may have turned a startup into a medium or large-sized company. It may have been completed in one continuous sweep, or the drug may have suffered long gaps. Whatever the case may be, each drug gives us many opportunities to learn about navigating tricky scientific and technical adversities. When drugs reach the market, unfortunately, all of this story behind those drugs gets forgotten or is only known by those who were directly involved in the R&D behind those drugs. For a drug discovery institute like us, really understanding the backstory is of immense value, because you never know, they might inspire ideas and help us find solutions to the problems that we work on. Today we have two awesome teams, and they will present the stories of resmetirom, which is approved for non-cirrhotic, non-alcoholic steatohepatitis, and suzetrigine, a non-opioid for moderate to severe acute pain. These drugs received the breakthrough therapy designation by the FDA because of their potential to impact human health. The student teams will take us through the fascinating journeys of these drugs. They have spent several weeks studying the literature and the patents and figuring out the key data that led to these drugs receiving regulatory approval. And you will see as they present. I do not wish to steal their thunder, so I'll say no more, but I do want to acknowledge and appreciate the student presenters for the day. Swecha Singh, Priyanka, Asta Singh, Neha Agarwal, Ardra Chakraborty, Lovely Dikshit, Ashutosh Sharma, and Stanzin Nurbo. Let's give them a hand. Of course, this event would simply not be possible without the mentors of the program. Dr. Amir Nazir, Dr. Anil Gaikwad, Dr. Kumaravelu, Dr. Prem N. Yadav, and Dr. Prem P. Yadav, and Dr. Ajay Srivastava. Thank you for your utmost dedication and hard work and for inspiring the students to do their best. Finally, thank you all for being here today. It means a lot to all of us who are involved in organizing the event. It means a lot more to the students who are presenting, your witness to their journey and their learning experience. And therefore, it's very special that all of you have taken time out to be with us. Thank you very much. I hand it back to Neha. Switcher, okay. Is it on?
Thank you so much, ma'am. Let's start our today's talk, Blueprint of a Breakthrough Inside the Development of resmetirom. Now we'll walk you through the journey of a revolutionary drug in liver disease. So my name is Swechha. I'm a second year graduate student working under the supervision of Dr. Siddharth Chopra from Molecular Microbiology and Immunology Division. Today, we delve into an exciting breakthrough in modern drug development, resmetirom, the first FDA-approved therapy for MASH. Let's begin by understanding why this drug marks a major milestone in liver disease management. Resmetirom, also known by its brand name Resdifra, was approved in the year 2024 on March 14 by FDA in an accelerated pathway. It is indicated for adults having F2 to F3 grade of fibrosis and is advised to be used along with diet and exercise. This innovation comes from Madrigal Pharmaceuticals and it's true first-in-class therapy for MASH. It is available in oral tablet form in 60, 80 and 100 mg of doses. MASH and MSLD are not rare. In fact, they affect a huge portion of global population. As a lifestyle-related disease, the numbers are increasing rapidly. It affects almost 30% of global population, and we can clearly see that India also falls in this range. The financial burden of MASH is staggering. It was estimated in the year 2024 that over 80 billion spent on healthcare costs related to MASH, and this number is expected to rise to 100 to 120 billion dollars per year by 2030. These costs include diagnosis, treatment, hospital stays, liver transplant, and productivity loss. This shows how urgent the need for treatment truly is. So resmetirom is approved for MASH. Now everybody is like, what is MASH? We know only one MASH, that is mashed potatoes. But this MASH is different from that. How? Let's hear it from the experts.
MASH is metabolic dysfunction associated steatohepatitis.
MASH is a term where there is chronic overnutrition, where the liver is storing these excess nutrients.
What ends up happening is you get both fat as well as inflammation in the liver. If you let that fat and inflammation go inside of the liver for long periods of time, that scar tissue starts to build up and can lead to cirrhosis of the liver,
which is a very severe condition and unfortunately a life-threatening situation.
For the majority of patients, the development of MASH appears to be very strongly linked to the development of metabolic syndrome. So patients who are either overweight, have high blood pressure, diabetes, their lipids are abnormal, or they tend to put weight more centrally on their abdomen instead of distributing it across their body, that predisposes them to also taking some fat and putting it instead inside of the liver.
Like many metabolic conditions, it is asymptomatic until it's too late. So that's why it's really important that whenever there are any indicators, and indicators may be things like blood tests or ultrasounds, that's taken seriously and we act.
This illustrates the massive at-risk population and need for early intervention. There are several factors that can accelerate MSLD to MASH progression. Food items that are rich in sugar, salt and saturated fatty acid leads to obesity, and obesity is directly linked to MASH. Now everybody is like, agar ye sab nahi khaye to kya khaye? So the food items that are green in color, we can have them. It will decrease the likelihood of MASH. It is essential to distinguish ALD, that is alcoholic liver disease, from MSLD. Although both affect liver function, but their primary cause is different. ALD is caused due to excessive alcohol intake, while MSLD is linked with metabolic syndrome. Let's see whether it is gender biased. So both are more common in males, but in case of MSLD, it increases with increasing the age of females. And what is the progression rate from steatohepatitis to cirrhosis? It is 3.2 to 12.2% per year in case of ALD and 1.3 to 3.5% per year in case of MSLD. The major outcomes includes SCC, that is hepatocellular carcinoma, liver infection and liver failure in ALD, and in MSLD it is cardiovascular events and hepatocellular carcinoma. Despite the growing burden, MASH and MSLD remains underdiagnosed and undertreated. The key unmet needs include limited availability of drug until resmetirom, lack of non-invasive diagnostic tools, low awareness among doctors and patients, issue with patient compliances, fragmented research and inconsistent data, which also highlights the variability in disease progression and treatment response, which complicates its management. So this is the pipeline of drugs that are under development for MSLD. Out of all this, resmetirom is the only one that receives full approval for treatment of MASH, while others are still in preclinical, clinical, or under regulatory reviews. This shows how much of a trailblazer resmetirom is in this therapeutic area. Now we'll explore the journey of resmetirom from its molecular design to clinical data. So I would like to call my fellow teammate Ms. Astha, working as a SARF under the supervision of Dr. Anil Gaikwad from Pharmacology Division.
Thank you, Sweksha, for introducing us to the concept of MASH and setting the stage for discussion. A very pleasant good afternoon to all of you. It's an honor to speak today on a topic that sits at the crossroad of hepatology, endocrinology, and the molecular medicine. That is from fatty liver to fibrosis, breaking the therapeutic void in MASH. Despite being long labeled as a silent pandemic of the 21st century, MFLD affects 1 in 4 people globally. Today, I will walk all of you through how does metabolic dysfunction fuel its progression from simple steatosis to fibrosis and beyond. Major metabolic causes of the MFLDs are the intake of the Western diet rich in the fructose and the sucrose, which promotes the hepatic de novo lipogenesis and the lipid accumulation in the liver. At the center lies the insulin resistance in the skeletal muscle and the adipose tissue. The third major cause is the dysfunctionality of the adipose tissue due to the overnutrition or the obesity, which promotes the MFLD through the several key mechanisms. So it can be said that MFLD is not just a liver disease, it is a hepatic manifestation of the systemic metabolic syndrome. Why do the people with fatty liver remain stable for the years while others silently progresses from simple steatosis to fibrosis and beyond? In the essence, we will move beyond the idea and the concept of the fatty liver as a benign and instead look MASH as a dynamic progressive disease. At the center lies the insulin resistance, overnutrition, or the intake of the Western diet, which promotes the hepatic de novo lipogenesis and the lipid accumulation in the liver and often lead to the NFLD, that is a non-alcoholic fatty liver disease. Due to the chronic inflammation and the injury, various immune cells get activated and lead to the steatosis stage characterized by lobular inflammation and the ballooning degeneration. Due to the excess lipotoxicity, endoplasmic reticulum stress, oxidative stress, hepatic stellate cells get activated, which are the major driver of fibrosis. And due to the chronic injury inflammation along with the fibrotic remodeling, epigenetic alterations and the genomic instability lead towards the hepatocellular carcinoma. So globally, 25% of the population is living with NFLD. Out of those, 20% generally progresses towards the NASH and 5 to 20% progresses towards the cirrhosis. The annual risk of developing the hepatocellular carcinoma is actually 5 to 13%. So the early stages, that is the NFLD without the severe fibrosis, are the reversible stage of this disorder, whereas the latter stages, that is the cirrhosis and the hepatocellular carcinoma, are the potentially irreversible stages of this disorder. Is the hypothyroidism a risk factor for non-alcoholic steatohepatitis? So recent evidences have suggested that the low serum thyroid hormone levels are associated with non-alcoholic fatty liver disease. The hypothalamus generally produces thyroid releasing hormone which acts on the pituitary gland to produce thyroid stimulating hormone which acts on the thyroid gland to stimulate the secretion of triiodothyronine and thyroxine hormone. So in the normal liver, this triiodothyronine is converted into the thyroxine hormone by DIO1 enzyme. But in the chronic or the injured liver, this triiodothyronine is converted into the reverse thyroxine hormone, reducing the bioavailability of the thyroxine hormone. Thus, the thyroid hormone plays a very important role in boosting the liver health. It generally promotes the hepatic lipogenesis, helps in the disintegration of the triglycerol assembly, promotes the lipophagy, and promotes the beta fatty acid oxidation genes. So why does the thyroid hormone receptor beta is an attractive target for the MASH treatment? Thyroid hormone receptor has two major isoforms. Thyroid hormone receptor alpha, that is mainly present on the heart and the skeletal muscles and the adipose tissue. So activating it may cause the tachycardia, that is an increased heartbeat, bone loss and the muscle catabolism, whereas thyroid hormone receptor beta is mainly present on the liver. So activating it may boost the hepatic fat burning without any unsystemic side effects. Therefore, our thyroid hormone receptor beta agonist resmetirom holds a promising target therapy for the MASH treatment. Now I would like to invite my chemistry colleague Ms. Priyanka, working as a CSIR PhD scholar under the supervision of Dr. Ajay Kumar Srivastava in the Division of Medicinal and Process Chemistry. Over to you, Priyanka.
Thank you, Asta, for walking us through the role of thyroid hormone in the progression and management of MASH. So far, we have seen why targeting thyroid hormone receptor beta made so much sense. The path from an idea to the pill is never linear. It is a chemical odyssey with include decades of exploration, countless molecules, failed attempts, lucky trials, and brilliant design. Resmetirom didn't just appear one day in lab. It is a result of evolving journey with each generation of molecule refining what came before. But before we dig deeper into the chemistry and design strategies, let's rewind a bit. Long before we understood the thyroid hormone at its molecular level, hypothyroidism was treated using the gland extracts of pigs and sheep. Although it was potent enough to save some lives, but it comes with lot of side effects. But as the science progressed, the question shifted. What exactly made these glands effective? And the answer finally came in year 1952 when the Dr. Rosalind Pitcher was working at the National Institute for Medical Research for the very first time established the chemical structure of active thyroid hormone T4 and T3. Soon after establishment of the chemical structure raised an important question. Are we able to design a molecule that target the only tissue we want but mimic the properties of T3 hormone? This question sparked the development of synthetic analogs across the world, and the first attempt was made in the year 1974 with the generation of molecule DIMIT. A group of scientists led by Dr. Jorgensen and his team, they tried to modify the naturally occurring T3 hormone to make the newer analogs. They replaced the 3 iodine atoms with the alkyl groups like methyl and isopropyl to study its effect. After this modification, they found out that all these modifications result in reduced hepatic monodeiodination from T4 to T3 conversion, as well as presence of this alkyl group increases the lipophilicity of the analogs and increases the free drug availability in the plasma. Dr. Jorgensen and group also established a very important fact that complete removal of the halogen from the analogs results in total loss of the biological activity. However, if we keep the combination of halogen with alkyl groups, it preserves the biological activity. DIMIT didn't work very well in the liver and ended up by causing hyperthyroidism in the newborn rat. So even though DIMIT didn't succeed, it gave our first real lesson that halogens were not just decorative. They are playing a very critical role in how the molecule is actually working. Nearly 15 years after the discovery of first molecule, researchers took the first efforts to design the selective molecules towards the beta isoform. A group of scientists led by Dr. Leeson, they tried to do the structural modification of the natural T3 hormone, especially at the 3' aryl methyl substituent, and they found out that presence of the electronegative group at the para position increases the receptor binding as well as the selectivity in the in vivo settings. The Dr. Leeson, they have screened more than 20 nitrogen-containing scaffolds, and out of which the selective thyromimetic activity being only observed in the case of the pyrazinone core. Although all these initial analogs didn't possess the sufficient pharmacokinetic and safety profile and therefore never advanced to the formal human trial. In retrospect, it is fascinating to realize the pyrazinone core, which is now known as a defining chemical feature of resmetirom, was chemically being explored back in year 1918 At that time, it didn't yield a clinical candidate, but definitely served as a structural template for the newer analogs with more precision and selectivity we ultimately needed. Moving forward, the 10 years of extensive research led us to the molecule GC1 in the year 1998, which was discovered in the Dr. Scanlon lab. Dr. Scanlon and team, they tried to perform some chemical modification in the natural T3 hormones, and they did 3 major chemical modifications. First one,
they replace all the iodine atoms with the alkyl groups to maintain the lipophilicity but avoid the bulk and reactivity of the iodine. Second, they replace a biaryl ether linkage in the core with the more flexible methylene linkage in order to establish the role of oxygen in governing the biological activity. And at the last, they replace the amino acid side chain with the oxyacetic acid to improve the polarity of the molecule. All this chemical modification collectively increases the 10 times more selectivity of the newer analogs for the TR beta receptor. GC1 did reach the phase 1 clinical trial but got discontinued because of the undesirable CNS side effects. From the synthetic chemistry point of view, working with such iodine-rich scaffold poses some hurdles. For the first one is the synthesis of the biaryl ether linkage is difficult because of the steric interaction caused by these large iodine atoms. And second one is the presence of 3 iodine atoms make the molecule highly prone towards the deiodination, especially in the acidic condition, which limits the further scope of functionalization on the aromatic ring. Now, the journey for finding the selective beta thyromimetic analogs took a very interesting turn in the year 2003. A group of scientists led by Dr. Robert Daw, they visited a class of forgotten molecules which were initially used as an anticoagulant in the Pfizer veterinary division designed by Max Miller back in year 1975. Dr. Daw observed a structural similarity between the 6-aza-uracils analog and the natural T3 hormone, and hence they decided to perform extensive structural modification to find out its effect on the TR beta affinity. After the modification, they find out that the removal of the acidic proton from the third position reduces the activity of newer analogs by 50-fold, as well as converting 6-aza-uracil to the uracil by removing nitrogen at the 6th position reduces the activity by 120-fold, as well as converting the aza-uracil to the urea result in total loss of thyroid receptor binding affinity. At the end, they established the fact that 6-aza-uracil based molecules showed 8 times more selectivity for the TR beta receptors. Sometimes the most potent innovations lie buried under the forgotten pharmacophore, and aza-uracil is a perfect example. The nucleus, which was overlooked back in 1975, was again reimagined, re-engineered, and formed the chemical core of the resmetirom. As we are walking through the chemical journey of the resmetirom, it is very important to mark the critical milestone, the patent filed by Hoffmann-La Roche in year 2007. It was the first time the molecule resmetirom or any chemical structure remarkably similar to it appeared first time in the literature. Under the patent section, they also mentioned these kinds of molecule could be used for the treatment of NASH. However, the synthetic approach provided by the patent suffered from lower yield, as well as the synthesis is not suitable for the scale-up cases. Now, as we reach the final chapter of this journey, we meet the molecule resmetirom, which was developed by Dr. Martha Kelly in collaboration of Madrigal Pharmaceuticals and Hoffmann-La Roche in year 2014. Dr. Kelly and her team took everything the field had learned over the decades to craft this molecule. They did extensive structural modification and tried to put each part of the molecule in order to maintain the same electronic configuration as of the natural T3 hormone. For that, they did some smart choices. Instead of replacing the iodine with alkyl group, as we have seen in the previous analogs, they replace it with the chlorine to maintain the lipophilicity. And the presence of the additional nitrile group shows interaction in the arginine 320 in binding pocket, which imparts 28-fold more selectivity of resmetirom toward the TR beta receptor. They also published a synthetic scale-up process for the synthesis of the resmetirom, with the overall yield ranging from 44 to 95%. Further, the preclinical and clinical studies will be covered by my other teammate, but before moving forward, we saw that the drug discovery isn't always about starting from scratch. Sometimes it's about seeing the old in a newer ways. Over to you, Astha. Thank you, Priyanka, for covering the chemical journey behind resmetirom. Resmetirom is a first-in-class oral active thyroid hormone receptor beta agonist. It generally acts on the thyroid hormone receptor beta present on the hepatocytes and enters the nucleus and activate the RXRR receptor and activate the uptake of various fatty acid oxidation genes such as CPT1 alpha, SREBF1, and promotes the de novo lipogenesis. Resmetirom is generally advised in the patients with or without early to moderate fibrosis, not typically to be used in the advanced cirrhosis stage. Resmetirom is designed to reduce hepatic steatosis, hepatic inflammation, and fibrosis. Worldwide studies using various hepatocyte cell line reveal that resmetirom reduces the lipid accumulation, activates the thyroid hormone receptor beta signaling, increases the hepatic fatty acid oxidation, decreases the de novo lipogenesis, enhances the mitochondrial biogenesis and the mitophagy, promotes the lipophagy, improves the cholesterol metabolism and the efflux, and gene modulation in the murine hepatocytes. RGS5, regulator of G protein signaling 5, a negative regulator of the GPCR, signaling is generally lowers the NASH progression. Then its activity is generally restored by resmetirom. RGS5 reactivation leads to the inactivation of various inflammatory pathways such as STAT3 and NF-kappa B signaling. Resmetirom reduces the inflammatory cytokine and the inflammatory progression in the MASH progression. Resmetirom reduces the hepatic stellate cell activation, a major driver of fibrosis. Diving into the in vivo efficacy of resmetirom, so in the diet-induced MASH model, HFD was given to the mice for 34 weeks and the resmetirom was administered at a dose of 3 mg per kg for 8 weeks, and the results were remarkable. It reduces the hepatic steatosis, reduces the plasma ALT activity, and reduces the liver weight. In the another composite MASH model, the HFD was given along with carbon tetrachloride, a chemical inducing agent for MASH, for 28 days, and resmetirom was administered at a dose of 3 mg per kg for 15 days. It significantly reduces the body weight, it reduces the liver weight, and it also reduces the hepatic steatosis. In the choline deficient HFD induced model, CDA-HFD diet was given for 1 week to induce MASH, and resmetirom was given at the dose of 10 mg per kg for 10 weeks. Resmetirom significantly reduces the body weight, reduces the plasma ALT and the AST activity, and reduces the hepatic steatosis. Let us briefly discuss the pharmacokinetics of the resmetirom. So, pharmacokinetic study generally reveals that how the drug is absorbed, distributed, metabolized, and eliminated from the body. Resmetirom is a first-in-class orally active thyroid hormone receptor beta agonist. After the post-oral feeding, it retains in the plasma for about 2 hours. Resmetirom shows a very good bioavailability of about 74%. The T half-life was found to be about 32 hours, suggesting a single dosing. It shows a major good protein binding affinity of about 99%, and it was majorly metabolized in the liver by hepatic CYP3A enzyme, a cytochrome P450 enzyme. The route of the excretion was found to be mainly through the fecal way. The drug-drug interaction study reveals that resmetirom is a CYP2C8 substrate as well as an organic anion polypeptide transporting substrate. Concomitant use with the moderate CYP2C8 inhibitors along with the inhibitors of OATPB1 and OATPB3 inhibitors may increase the resmetirom plasma concentration and AUC. Resmetirom increases the plasma concentrations of some statins such as atorvastatin, pravastatin, rosuvastatin, and simvastatin, which may increase the risk of adverse reactions related to these drugs. Diving into the preclinical toxicology profile of the resmetirom, so 28 days and 90 days toxicity studies in the rat shows no target organ toxicity. High doses in the juvenile rat show reversible effect on the bone growth. No evidence of the genotoxicity were reported. No teratogenic effects were reported. Supratherapeutic doses called mild reversible thyroid hormone axis changes and no malignancies were observed. Before entering into the clinical trials, a series of IND-enabling studies were conducted which reveals that resmetirom showed no significant effect on the cardiovascular, central nervous system or respiratory systems at the therapeutic dose. No dose-limiting toxicities were identified at the clinical exposure level. The comprehensive preclinical data strongly supported the IND filing and the rapid progression into the human clinical trials. Now I would like to invite my colleague Neha Agrawal, working as a CSIR-SR 5th year under the supervision of Dr. Prabhat Ranjan Mishra in the Division of Pharmaceutics and Pharmacokinetics. Over to you, Neha.
Having understood the chemical profile and the encouraging preclinical outcomes of resmetirom, let's now move to the heart of its translational journey, the clinical data. Good afternoon, everyone. Today, I will walk you through the sequence of clinical trials, starting from phase 1 to the pivotal phase 3 outcomes that ultimately led to the FDA approval. Now, before we begin, let's take a quick glimpse at the major milestones in the clinical development of resmetirom. In 2016, Madrigal Pharmaceuticals filed an investigational new drug application, marking the formal initiation of the clinical development. Shortly after which, phase 1 trials began, leading to phase 2 trials between 2017 and 2019. Based on the progress of phase 2 trials, 2 major phase 3 trials were conducted in 2020. Then, a new drug application was filed with the US FDA in June 2023, which received the priority review status. And finally, in March 2024, resmetirom received the FDA approval for treatment of NASH, marking a turning point in the treatment of this complex liver disease. So, let's now look at each of the phases one by one. The phase 1 trial involved healthy individuals and patients with hepatic impairment, and the aim of the study was to assess the pharmacokinetics, pharmacodynamics, safety, and tolerance of resmetirom in the dose range of 50 to 200 milligrams. It was found that in healthy individuals, resmetirom showed a dose-proportional pharmacokinetics, short half-life, and no food effect, making it suitable for one's daily use. From pharmacodynamic perspective, it showed early lipid-lowering activity, and most importantly, there were no severe side effects observed. In phase 2, 125 biopsy-confirmed NASH patients were enrolled, and the objective of this study was to assess reduction in hepatic fat, liver enzymes, and safety at the end of 36-week time period. So at the end of 36 weeks, it was found that in resmetirom-treated patients, there was 37.3% reduction in the hepatic fat as compared to only 8.5% reduction in the placebo group. Also, resmetirom treatment led to reduction in liver enzymes such as ALT and GGT, showing improvement in hepatic injury and hepatic inflammation. Again, there were no serious side effects observed in this phase, and the data allowed progression to the next phase of the clinical trial. There were 2 major studies conducted as a part of phase 3 clinical trial. The first one was to evaluate the safety and biomarkers of resmetirom in non-alcoholic fatty liver disease patients, the findings of which were published in Nature Medicine. Selection of participants is the first and the most important step while performing a clinical trial. To become a part of this trial, patients needed to cross the 18 years age bar and provide a written informed consent. Also, they needed to be suspected or confirmed with NASH with a fat fraction of more than 8% during the screening period with a stable dyslipidemia therapy for more than 30 days. Also, patients who were regularly using drugs historically associated with NAFLD could not be enrolled in this trial. Neither could the patients diagnosed with hepatocellular carcinoma, hepatic decompensation, chronic liver disease, active autoimmune disease, uncontrolled hypertension, or any other active serious medical condition with a likely life expectancy of less than 2 years become a part of this trial. So, after all the screening, 1,143 patients were randomly divided into 4 groups: double-blind placebo, double-blind resmetirom 80 mg, double-blind resmetirom 100 mg, and open-label resmetirom 100 mg. The duration of the study was 52 weeks, and the primary endpoint was to observe the incidence of treatment-emergent adverse effects, secondary endpoint being observation of lipid profile, hepatic fat, and liver stiffness at the end of 52 weeks. So at the end of 52-week time period, it was found that resmetirom was well tolerated at both 80 mg and 100 mg doses, and there were no serious treatment emergent adverse effects in the resmetirom group as compared to the placebo group. Resmetirom treatment also led to significant reduction in LDL cholesterol, apolipoprotein B, and triglycerides from baseline as compared to the placebo. A non-invasive technique, that is magnetic resonance imaging proton density fat fraction technique, was used to assess the hepatic fat. And at the end of week 52, it was found that there was significant reduction in the hepatic fat in the resmetirom treated arm as compared to the placebo. Markers of liver injury such as alanine aminotransferase, aspartate aminotransferase, and gamma-glutamyl transferase were also significantly reduced in the resmetirom arm as compared to the placebo. Interestingly, there was an increase in the sex hormone binding globulin in the resmetirom arm as compared to the placebo. Now, this increase in sex hormone binding globulin correlates with the THR beta activation and also reflects the resmetirom exposure. The second study done as a part of clinical trial phase 3 was done to evaluate the efficacy and safety of resmetirom in patients with NASH and fibrosis, and the findings of which were published in the New England Journal of Medicine. The inclusion criteria was very similar to the previous one, except to become a part of this trial, patients needed to be confirmed with NASH with fibrosis with characteristics such as steatosis, ballooning degeneration, and lobular inflammation. The exclusion criteria was same as the previous one. So after all the screening, 966 patients were randomly divided into 3 groups: placebo, resmetirom 100 mg, and resmetirom 80 mg. Although the actual duration of the study is 54 months, but the outcomes observed at a surrogate endpoint of 52 weeks were considered for the approval. The primary endpoint was to observe NASH resolution and improvement in fibrosis by at least one stage with no worsening of the NAFLD activity. And the secondary endpoint was to observe the lipid profiles, liver enzymes, and safety in the patients. So, at the end of the study, it was found that NASH resolution with no worsening of fibrosis was achieved in 25.9% of the patients in the 80 mg resmetirom group and 29.9% in the 100 mg resmetirom group, as compared to only 9.7% of those in the placebo group. Fibrosis improvement by at least one stage with no worsening of the NAFLD score was achieved in 24.2% of the patients in the 80 mg resmetirom group, 25.9% in the 100 mg resmetirom group, and only 14.2% of those in the placebo group. Now, apart from the histological findings, resmetirom also led to reduction in the LDL cholesterol level and the liver enzymes as compared to the placebo group. Non-invasive techniques such as MRI-PDFF and FibroScan outcomes corroborated with the histological findings, showing significant reduction in the hepatic fat with continued resmetirom treatment. The safety profile was very similar as compared to the previous phases. Diarrhea and nausea were more common but very mild, and the other adverse effects were comparable in all the 3 treatment groups. So finally, after all these extensive studies, in March 2024, Madrigal Pharmaceuticals received the FDA approval for resmetirom for treatment of NASH. The FDA approval for resmetirom for the treatment of NASH with liver fibrosis not only marks the first approved treatment for NASH, but also proves to be a turning point for patients who earlier had no pharmacological options. It shows how decades of scientific innovation, clinical rigor, and regulatory alignment can come together to bring a novel therapy from bench to bedside. The development and regulatory submission of resmetirom followed a rapid timeline, indicative of its breakthrough status and the urgent unmet need it addresses. When the Madrigal Pharmaceuticals filed a new drug application with the US FDA, it received a priority review status. Priority review status is given to those applications for drugs that, if approved, would lead to significant improvements in the efficacy and safety of treatment, diagnosis, and prevention of severe conditions. Notably, the FDA did not convene any special advisory committee meeting to discuss this application, a decision that clearly indicates a compelling risk-benefit profile of the submitted data. The absence of such a meeting suggests that the internal review divisions of FDA found the submitted data to be sufficiently robust and the overall profile of the drug to be very straightforward so that it can be given an accelerated approval. Although resmetirom has been given the approval for use in real world, it still needs to complete the 54-month-long ongoing phase 3 study. And apart from that, it needs to complete the post-marketing commitments as a part of accelerated approval, such as they need to perform a safety, PK, and efficacy study in prepubertal and postpubertal patients. They need to study the effect of resmetirom in pregnant females and lactating mothers so as to assess any fetal or maternal complications. They need to conduct a PK study in patients with impaired renal function. A clinical drug-drug interaction study using resmetirom and organic anion transporting peptide also needs to be conducted. And lastly, they need to perform an environmental fate and ecotoxicity study and submit the final report to US FDA. If the post-marketing studies fail to verify the clinical benefit or are not performed with due diligence, the FDA may withdraw this application. Thus, the story of resmetirom is a remarkable example of translational science at its best, where a well-defined hypothesis led to the development of a liver-targeted thyroid hormone receptor beta compound, whose journey was carried forward by preclinical validation and clinical development. And as we discuss this achievement, it further opens the door for future drug development in hepatic and metabolic disease landscape. With this, we now come to the part I have been most excited to talk about: Picrolib drug, an indigenous solution for NFLD developed right here at CSIR-CDRI Lucknow, one of the most prestigious drug discovery and drug development institutes of our country. It has been derived from the traditional medicinal plant Kutki, botanically known as Picorrhiza curua, which has been long revered in Ayurveda for its hepatoprotective properties. It targets NAFLD and aims to prevent the progression to severe conditions such as liver cancer and liver cirrhosis. It has been developed under the phytopharmaceutical guidelines laid down by DCGI and has already advanced to phase 3 clinical trials being conducted across various parts of our country. If successful, it would be the first phytopharmaceutical NAFLD drug from India, setting a benchmark in the field of public health and innovation sector. So let us all take pride in the fact that Indian science is not only conserving traditional knowledge, but is also transforming it into globally beneficial therapeutic options, keeping both safety and efficacy at the forefront. With this, I conclude this session and thank you all for your kind presence and immense patience. We will reconvene in 15 minutes for the next session.
So meanwhile we are waiting for the tea. I request everybody to be seated here only. Tea is not available. So if you have any questions, if you want to discuss something regarding this, we can have some questions. Meanwhile, we can have some discussion. Like there are people from outside institute, basically from the pharma colleges. Yeah, please. You wanted to ask something? So if you have any questions, so I must appreciate that these people have, like, all of the presenters, please be on the stage. Please come here, yeah, everybody of you. So at the moment it is not available in India. It is clinically approved only in US FDA, and at the moment they have sufficient capacities to provide there in the US. It is CDSCO has not given approval per se for India unless until there are certain the documentation has to be filed for Indian approval also. So at the moment it is not approved. So we have not dwell into the cost aspect yet, because it is very recently approved, just approved in the March 24. So even in US also, for providing them to the all sites, at the moment they have limited capacities. They are working on it. Presenters, be on the stage.
Pricing in INR comes out 88,000 for 30 tablets. So it's on a high side.
Please be on the stage.
That's an
I invite
all four dollar pricing. Dollar pricing
So thank you, team. You must realize that this whole whatever work has been done by them in last 15 to 20 days, and they have been working not only getting all the information, putting all the information, they have to go under rigorous comments from all of us mentors, and even I must appreciate Dr. Radha has also looked into the presentation, director has also looked into the presentation 2 to 3 times, and she has also given her inputs. So every input was there and they had to do all hard work. Thank you so much for putting in the best show. And what I wish to say that it is an experience for the pharma people coming from outside. You could have realized that it is not easy to bring the drug to the market. It is like, putting all hard efforts for 20 to 25 years, and story starts somewhere else and it leads to the drug. Eventually a long journey has to go through. So if you have any questions, please open up and ask.
Would you like to talk about the process and how?
Yeah, you can come.
So it was a very amazing experience because all the mentors supported us in each and every stage of this progression, and I want to thank all the mentors and especially ma'am, director ma'am, to support us and to start this symposium at this level. Thank you.
Hello, everyone. So, CSIR-CDRI, I would like to say, is not only a typical research institute, what I have felt in my journey here. So, it gives you various opportunities to, look at your skills, develop your skills, what you are good at, maybe it art, painting, poster development, oratory skills, anything. So, it depends on you, how you are taking those opportunities and how you are utilizing them. It's fun to, sometimes step outside the lab, do something else which is not completely haywire. It's related to science, but a little different from the bench work. So thank you, ma'am, for always pushing us and motivating us to do something new. And this experience was really great because somewhere all of us sitting here are contributing our bit in the process of drug development, but we do not get to know the complete picture. I think through today's presentation you'll get to know what is the complete picture, starting from scratch to the product in the market, which is very important. A single pill which we swallow, as we were discussing with director ma'am in the morning, a single pill which we swallow, it takes a hard, like literally a big man force behind that. Lots of hard work, decades of hard work, so many people working day and night, failed experiments, small successes, and that makes a one single pill which we just swallow casually. So we should, give credit to all the hard work, admire the hard work, the long process journey which goes behind everything. This is all I would like to say.
I would like to thank Director Ma'am for starting this new symposium series. As coming from the pharmacy background, when I'm exposed to this topic, I was very excited to see the journey behind the drug coming into the market. While I was looking for the literature, I found out that it's not only a 2, 3, or 10 years of research. As you already have seen, all these scaffolds, they are being explored back in years, then nobody put focus on it, then again somebody saw the same thing with a different perspective, and all this thing will bring a drug into the market. So I would say it's really amazing to finally realize that every time we stress to discover something very new which is novel, nobody have reported it, but sometimes we just focus, or we should change the way we are seeing the old thing. So it was a very great experience. I have learned a lot. Thank you so much, ma'am, for giving this opportunity. Thank you.
And I must appreciate Pacha because he is from the infection biology background and she is talking into the metabolism.
As everything is said by my teammates, I just want to thank you, everyone. Thank you, Director Ma'am. Thank you, my mentors, for giving us this chance to full flourish, give this presentation and everything. Thank you so much, everyone.
So, attention span is only around 40 to 45 minutes, and you need to have a break before that. And let's grab a cup of tea and come back within 15-20 minutes. We'll have another session. 3:15. 3:15. You have to be back. Thank you.
So, good afternoon, everyone. I hope you all enjoyed the first phase of today's program on innovations in medicine, where we explored the fascinating journey of resmetirom. For our second phase, we'll be going through the story of suzetrigine, a drug that tackles a problem that every one of us has faced at some point in our lives, pain. Not the emotional kind, but the physical manifestation, one that can reduce the quality of life and challenge even the strongest amongst us. So, let me welcome you all to the story of suzetrigine, which will be presented by four of us, right from its conception, which will be covered by me, Ardra, to the chemical composition presented by Lovely, followed by Ashutosh with the mechanistic aspects and preclinical data, and rounded up by Stanzin with the clinical studies. So with this, welcome to the story of suzetrigine. A novel therapeutic journey begins. Suzetrigine, also known by the compound name VX548, is the first in its class non-opioid analgesic which has been developed by Vertex Pharmaceuticals. This small molecule has an impressive CV. It affects the sodium channel NAV with remarkable selectivity. Suzetrigine has been approved by the FDA on January 30th of this year, and it will be marketed as oral tablets under the brand name of Journavax. Suzetrigine has been approved for the treatment of moderate to severe acute pain. Pain is a sensory concept which can be divided into either acute pain, which is short-lived, or chronic pain, which lasts for 3 months or longer and may signify some underlying medical conditions. Our drug of interest, suzetrigine, deals with the mitigation of acute pain, which, if left untreated for a long time, may ultimately lead to chronic pain. Before we delve deeper, a reality check. Pain is a global affliction, and in India there are millions who suffer from chronic pain. This includes, but is not limited to, pain due to arthritis, neuropathy, post-surgical, post-traumatic malignancy, and so on. Now, out of these types, other than neuropathic pain, which occurs due to nerve damage, most other types of pain may be classified as nociceptive pain, which arises due to tissue damage. Therefore, to better understand the science behind suzetrigine, let us first understand the nociception or pain pathway. So, when the body receives a damaging or noxious stimulus, the signals are carried through to the dorsal root ganglion or the DRG neuron, and this process is known as transduction. From here, the signal is carried to the dorsal horn of the spinal cord up through the spinothalamic tract to the thalamus and then to the somatosensory cortex of the brain, a process known as transmission. Once in the somatosensory cortex, the brain creates a perception of pain and subsequently sends signals for its modulation. Now, along this pathway, there have been various druggable targets which have already been explored for the mitigation of pain, which are as follows. Currently existing drugs that have been approved by the FDA for pain treatment mainly fall into one of these broad groups, namely NSAIDs, COX-2 inhibitors, acetaminophen, NMDA antagonists, and so on. However, the continued usage of these drugs can lead to various side effects such as hepatotoxicity, renal toxicity, increased cardiovascular risk, GI bleeding, and so on. Perhaps the most harmful of them all are opioids. Opioids still continue to be routinely prescribed because of their potency in pain relief. However, the long-term usage of opioids can lead to addiction, tolerance, and severe withdrawal symptoms if usage is discontinued. So severe is the opioid epidemic that in America, there are approximately 25 deaths per 100,000 of the population due to opioid overdose, many of which are due to commonly prescribed opioids. From this data, it is evident that there is an unmet need for non-opioid based safer alternatives. This led scientists to stumble upon an interesting story, that of a small young 10-year-old boy who used to perform dangerous street stunts like walking on hot burning coal and doing self-mutilating behavior like inserting knives throughout his body, but he didn't feel pain in the slightest. How was he able to do it? A team of scientists led by Dr. Geoffrey Woods at the University of Cambridge found that he had a mutation in this gene, the SCN9A gene, that led to a mutated voltage-gated sodium channel or NAV channel, and because of this mutated channel, there was no pain signal which was being relayed to his brain and consequently blocked pain perception. Therefore, this led scientists to ponder, Could these clinical findings be translated into therapeutic benefits through the exploration of new pain management targets? So before new pain management targets could be explored, it was important to know more about these NAV channels. So remember the nociceptive pathway that I showed you a few slides back? Well, once the body receives the nociceptive stimulus, this signal is passed with the help of these NAV channels present on sensory neurons through the generation of action potential. Now, different types of NAV channels are present in different areas of the body, with NAV 1.7, 1.8 and 1.9 predominantly found in the peripheral nervous system or PNS. As can be seen here, NAV 1.9 is mainly responsible for maintaining the neuronal resting membrane potential. NAV 1.7 contributes to the threshold potential, and NAV 1.8 is the major contributor to the rising phase. Both NAV 1.7 and NAV 1.8 were considered as potential pain targets. However, NAV 1.7 also showed some off-target effects since it is also expressed in the CNS. Hence, scientists decided to focus their attention on NAV 1.8. So, based on both clinical studies as well as laboratory data, scientists saw that NAV 1.8, when there was a gain of function mutation in these NAV 1.8 channels, it leads to hyperexcitability, or the subject feels extreme pain even at the slightest of stimulus. Conversely, when there is a loss of function mutation in NAV 1.8, it leads to hypoexcitability or no pain sensation at all. This led scientists to conclude that NAV 1.8 could be considered as an important druggable target for pain because of it was already validated in both in vitro and in vivo studies because of minimal off-target effects, easy designing of drug targets, and it would provide a safer, non-addictive alternative to opioids. So therefore, leveraging the critical role of NAV 1.8 in pain management, suzetrigine became the first potent non-opioid painkiller to be approved by the FDA. Because of its safety profile, patient tolerability, and effectiveness, suzetrigine was recognized as the 2024 Molecule of the Year. Here is a brief timeline of the development of the drug. So, suzetrigine marks a significant milestone in pain development due to its novel mechanism of action with the capacity to transform the lives of millions worldwide. It is a paradigm shift in neuroscience and pharmacological studies because this can not only is improving lives currently, but it also has the potential to fundamentally reshape how we treat pain in the future. And with that, I would like to call upon my friend Lovely to present the chemical part of this. And thank you.
Good afternoon, everyone. So first of all, I would like to thank my colleague Ardra for setting up the stage. So not taking much of the time, I would like to begin the presentation entitled From Molecule to Medicine, the Chemistry Behind Highly Selective Suzetrigine. Who thought that a small drop of venom can entirely silence the central nervous system? These toxins hijack the sodium channels while disrupting the electrical rhythm of life itself. Insight gained from these toxins helps in the development of various novel pharmaceutical agents, especially working in the management of pain. The first documented use of sodium channel blocker was for neuropathic pain was cocaine injection, and its use was reported in year 1884. Soon, subsequent research was found that these cocaine injections were causing severe side effects. Synthetic chemistry research was already ongoing in various chemical laboratories that marked a serendipitous observation that various anticonvulsants, antidepressants, and antiarrhythmics were helping in treating several pain-like conditions in humans. But the challenging part was to achieve the high selectivity towards NAV channel, because all these local anesthetics present at that moment of time were highly non-selective and causing severe off-target effects.
So by early 2000s, Abbott, Icogen and Pfizer emerged as early pioneers in the NaV1.8 inhibitor research field, initiating the discovery phase for NaV1.8 inhibitors, where Abbott and Icogen emerged as the first industry to come up with the very first NaV1.8 inhibitor that was selectively inhibiting NaV1.8 with IC58 nanomolar, and this particular molecule was 100 times more selective towards other NaV subtypes. But unfortunately, this molecule could not make up to the market. In a similar fashion, Vertex and GSK were also working together to come up with the molecule, and by the year 2009, they have already come up with the molecule VX409. But unfortunately, when the molecule was about to enter its preclinical development, the companies parted their ways, and no clinical insights and structural binding details were revealed by the group. In a similar fashion, there were other groups as well that were working together to develop voltage-gated inhibitors, and they are also in the race where their molecules are either in preclinical or clinical development. But the turning point came in year 2001 when Vertex acquisited Aurora. By the acquisition of Aurora Biosciences by the Vertex, the Aurora Biosciences had special expertise in ion channel chemistries and high throughput screening. Vertex took the advantage of that and laid its groundwork for the Vertex Brain program. And by the year 2025, Vertex has already cracked the molecule suzetrigine, which is now approved by FDA. So now we will look at the binding sites for the NaV1.8 inhibitors. Before jumping onto the suzetrigine, we will look at the other molecules that were there in the picture. So if we look on these molecules, that what were the binding sites available in the NaV channel for these molecules? These molecules are binding to the central pore domain region of this channel. So, but a subsequent study later revealed that the direct inhibition of the central pore domain region of this channel is causing severe side effects. Later, two other molecules came into picture, that is suzetrigine, that is LTGO33, developed by the Latigo Biotherapeutics, and our very own suzetrigine. So, a study was conducted for both these molecules, that what were the binding site for these two molecules, because the chemical basic skeleton structure for these two molecules seems to be quite similar. A mutagenesis study and engineered chimera studies were performed, and it was revealed that LTGO33 was binding to this allosteric site instead of this central pore domain region of the channel. And later, in case of suzetrigine, it was a domain swapping study was conducted, which resulted that this suzetrigine is also binding to the allosteric site of the channel. During the domain swapping studies, a very unique amino acid sequence was analyzed, that is this KKGS, and it was found that this presence of this amino acid is highly crucial for the selectivity of suzetrigine. Later, the detailed discussion of the domain swapping studies will be discussed by my friend Ashutosh. Now, suzetrigine is not something that was synthesized overnight. It had required a hard work. Vertex has continuously worked hard in the stream to get this molecule suzetrigine, and the journey begins by the year 2007 when the company published the very first patent with the molecule claiming to be with certain set of molecules with IC50 less than 2 micromolar. The company continuously kept revolving around these molecules and explored these molecules, and then ultimately the turning point came in year 2018 when they first launched a molecule VX150. This particular molecule was highly promising and showed very promising results in trials, but unfortunately this molecule due to severe side effect property, it was terminated from phase 2 trials, and the company didn't stop there. It kept revolving around different molecules, and by the year 2024 they came up with another molecule, that is VX548, and this molecule was later approved by FDA in year 2025. Now we will look on what were the major optimization that were performed on this particular molecule to develop the suzetrigine. So if we look on this molecule, we can see that there are there were many changes that were performed in this molecule, but the two major changes that were highlighted is first the introduction of this pyridine carboxamide side chain by removal of this 2-oxopyridine methyl phosphate group, and second one is the installation of this chiral group. Here instead of the six-membered benzene ring, they have introduced a 5-membered furan ring with 4 different stereo centers. So if we compare both these molecules, we can see that the IC50 in case of VX150, which was 86 nanomolar, and in case of Szytrazine, it was 0.75 nanomolar, which makes the molecule very extremely potent. And if we compare the selectivity in terms of selectivity, Szytrazine selectivity has increased to 31,000 times, which is a big number. And the two other problems that were also analyzed in case of VX150, first one was the severe side effects in case of VX150, and the another one was the high dosage problem. So in case when VX548 was analyzed, it was found that both these problems were resolved, and there were no severe side effects were seen in case of Szytrazine, and the dosage values were also reduced from 1500 mg to 100 mg. So now, what makes this molecule so special? The molecule is very special in itself just because of the introduction of this chiral, a 5-membered chiral center, a chiral ring. If we see, this molecule has at every 4 points, the molecule has a different set of stereochemistry, which makes this molecule extremely potent. But as soon as the stereochemistry at any point is changed, the IC50 has crashed down from nanomolars to micromolars, to the point that other isoforms of this molecule are not even active. So now we will come to the synthesis part of the Szytrazine. So if we look on this molecule, though the molecule seems to be quite smaller, but its synthetic journey is not easy. It requires a lot of hard work and perseverance. So if we focus on this molecule, it contains two key fragments. The first one is the pyridine carboxamide side chain, and the other is the trifluoromethylxylane core ring. So in the trifluoromethylxylane core ring is synthesized in the very first step, which is the mixture 5:1 diastero mixture, and later the pyridine carboxamide side chain ring is introduced in a set of steps by the, if we see the reaction mechanism synthesis, we can see that it is preceded with the reduction of this lactone ring, followed by the introduction of this protecting group, that is the 4-nitrobenzoyl chloride, followed by the introduction of this cyano group with the help of this TMSN trimethylsilane and in the presence of BF3 ethylate. Later, chiral auxiliary is introduced in the synthesis, so to keep the stereochemistry of the molecule maintained, and ultimately they have done the methanolysis while washing the molecule with methanol and ammonia, and they have reached to Szytrazine. So in earlier patents, the synthesis was not easy. It required more number of steps, and the yield was very less, as around 50 to 55%. Later, this patent was published in year 2024 with reduced number of steps and increasing the yield up to 95%. So the challenges that were involved in the previous synthesis mechanism of Szytrazine were the diastereomer formation and separation and the stereochemical selectivity, which are being resolved in this part. So here I would like to end my part and would like to call my friend Ashutosh to throw some light on preclinical data. Thank you.
So good afternoon everyone. So before starting my presentation, I would like to thank Lovely for giving insights on the chemical development of Szytrazine. Now I'll talk about the preclinical pharmacology and the mechanism of action of Szytrazine. So the preclinical pharmacology and mechanism of action findings were published by the Vertex Pharma Group in January 2025 in the journal Pain and Therapy. Now before coming to the findings, let's discuss in brief about the NAV 1.8 channel structure and the functions. So NAV 1.8 channel, it's a transmembrane protein which is predominantly expressed in the peripheral nervous system, and it's responsible for the pain signaling. So the molecular structure of NAV 1.8 channel comprises of 4 homodomains, and each homodomain comprises of 6 transmembrane helices, where the S4 helix acts as the voltage sensor domain due to the presence of basic amino acids, whereas the S5 S6 domain acts as the pore domains, which allows the conductance of sodium ions. Also the D3 D4 intracellular loop acts as the inactivation gate. So this was about the molecular structure. Now coming to the functioning of NAV 1.8 channel, so they are present in 3 physiological states: closed, open and inactivated. When the channel is at resting state or at resting membrane potential, the channel is closed and there is no influx of sodium ions, whereas upon the depolarization or increase in the positive voltages, it leads to the opening of the channel gates, which allows the entry of sodium ions. And upon the further depolarization, just to avoid the excessive entry of sodium ions, inactivation gates are now closed. This mechanism can also be studied by doing patch clamp electrophysiology, in which when the channels are at resting state, there is no any sodium current, and upon the depolarization, a fast sodium inward current can be seen, and subsequently after the channels get inactivated, current again becomes zero. So why this NAV 1.8 channel is important? Because the entry of sodium ions through these NAV 1.8 channels amplifies the pain signals in the pain conditions. Now the suzitrazine potency was studied by doing the patch clamp electrophysiology, which is considered as the gold standard technique for studying ion channel biology. So these are the membrane currents of NAV 1.8 channel expressed in HEK293 cells, and as you can all see, the application of 10 nanomolar suzitrazine inhibits both the peak phase as well as the sustained phase of the NAV 1.8 currents. Also, the dose response curve shows the sub-nanomolar potency of suzitrazine on NAV 1.8 channel when compared with all other NAV subtypes, with a greater than 31,000-fold selectivity ratio of NAV 1.8. To assess the potency of suzitrazine across the various species, their DRG neurons were isolated, and it was found that suzitrazine was found to be most potent in human DRG neurons with the IC50 value of 0.68 nanomolar, whereas the least potency was found to be in dog DRG neurons with IC50 of 740 nanomolar. So we can say that suzitrazine has a greater than 31,000-fold selectivity ratio for NAV 1.8 channel, and its highest potency for human DRG neurons makes it a suitable candidate for clinical studies. Now, after studying the selectivity and potency of suzitrazine on NAV 1.8 channel, next question comes that where does the suzitrazine binds? So this was experimentally confirmed by doing the domain swapping strategies, in which all of the 4 voltage sensor domains 1 to 4 of NAV 1.8 were introduced into the NAV 1.2 channel. So here you can see the NAV 1.2 membrane currents, and it was found that the application of suzitrazine had no inhibition on the sodium currents. Subsequently, when they introduced the VSD1 of NAV 1.8, VSD3 of NAV 1.8, and also the VSD4 of NAV 1.8 into the NAV 1.2 channel, suzitrazine had no inhibition. But interestingly, when the NAV 1.8 VSD2 domain was inserted into the NAV 1.2 channel, now suzitrazine inhibited the sodium currents, which confirmed the binding site of suzitrazine on the NAV 1.8 VSD2 domain. Now, further sequence alignment studies also suggested the unique KKGS sequence in the human NAV 1.8 VSD2 domain, which was experimentally confirmed when it was incorporated into the NAV 1.2 VSD2 motif, and it was found that again suzitrazine showed the inhibition. So suzitrazine binds specifically to the KKGS motif in the voltage sensor domain 2 of NAV 1.8 channel. Now, after studying the selectivity, potency, and the binding site of suzitrazine on NAV 1.8 channel, next question comes at how does suzitrazine modulates the NAV 1.8 channel. So to study that, let's discuss in brief about the types of NAV 1.8 inhibitors and how do they modulate the channel activity. So there are 2 types of NAV 1.8 inhibitors, pore blockers and the allosteric modulators. So pore blockers, as you can all see, they obstructs the channel pore, which prevents the entry of sodium ions, whereas the allosteric modulators, they don't bind on the channel's pore and they bind outside the channel pore into some allosteric sites, which changes the channel conformation, making it in a more closed state or reduces its probability to open. Now the mechanism can be studied by patch clamp currents. So here you can see these are the membrane currents before addition of pore blocker, and after the addition of pore blocker, the mean peak current amplitude decreases gradually with the increase in frequency stimulation, because increase in the frequency stimulation is leading to the more channel opening, and more channel opening increases the likelihood of this blocker binding more to the pore domain. Whereas when the frequency of stimulation is again decreased, the current inhibition by blocker is also decreasing. This is why, because the long interval between the 2 frequency stimulation is now leading to the dissociation of this blocker from the pore domain. Now allosteric modulators, unlike the pore blockers, irrespective of the frequency stimulations, their drug inhibition, their current inhibition is same from the very first pulse to the last pulse, and it is also called as the tonic inhibition. So the classical example of pore blockers are local anesthetics like lidocaine and tetracaine, whereas the example of allosteric modulators are LT033 and the drug of the day suzitrazine. So the local anesthetics, they are very much efficacious in pain reduction, but they have so many off-target activities because they bind non-selectively to the most of the voltage-gated sodium channels because of the structural similarity in the pore domains, and they have abrupt neuronal firing and neuronal signaling effects, whereas allosteric modulators, since they bind to the unique allosteric sites which are very much less conserved, so they have more targeted and less off-target activity. Now let's discuss in more about the suzetrigine mode of inhibition. So suzetrigine is a first-in-class allosteric modulator of NaV 1.8 channel, and it binds to the VSD2 domain of the NaV 1.8 channel, which stabilizes its closed state. And how this was confirmed? So this was confirmed by doing the patch clamp electrophysiology protocols, in which when the hyperpolarizing pre-pulse voltages were given, suzetrigine inhibited the channel in the dose-dependent manner, because hyperpolarizing pre-pulse voltages drives channel into the more closed state, and as we know that the suzetrigine stabilizes the closed state of the channel, so its inhibition is in the hyperpolarizing pre-pulse voltages, whereas in the case of pore blockers, they show their inhibition in the depolarizing pre-pulse voltages, because these depolarizing pre-pulse voltages drives channel into more open and inactivated states. Also, suzetrigine was found to be efficacious under the repetitive depolarization stimulations, which mimics the action potentials or the pain signals in pain sensing neurons, and it was found that the application of 10 nanomolar suzetrigine inhibited the current amplitude from the very first pulse to the last pulse, obviously, which is the tonic inhibition. And once suzetrigine binds to the channel, it stabilizes the closed state and won't lead channel to open again. So traditional rodent pain models are not very much efficacious in the clinical translation. So to assess the potential of suzetrigine for clinical pain reduction, isolated DRG neurons were used to record action potentials, and it was found that the application of 10 nanomolar suzetrigine also inhibited the action potential's peak amplitude significantly. So we can say that suzetrigine is an allosteric channel inhibitor, unlike channel blockers or pore blockers, and it maintains consistent inhibition over a wide range of voltages and action potential frequencies that could occur across pain states. Now, as we know, traditional opioid pain medications carry a risk of dependence, tolerance, and addictive potential due to their binding effects on central nervous system. So to rule out any evidence of addictive potential of suzetrigine, withdrawal studies were performed in the rats, where the drug was administered for continuously 30 days, and after 30 days drug administration was discontinued, and the body weight and the body temperature parameters were recorded for the withdrawal studies, and it was found that after the abrupt cessation of suzetrigine, there is no any decrease in both the body weight as well as the body temperature parameters, whereas the positive control group morphine, which is an opioid which develops addictive potential, was found to have a decrement in both body weight and the body temperature. So suzetrigine did not produce any signs of dependence at a pharmacology relevant exposures following the cessation of drug treatment. Also, the evidence for lack of abuse potential with suzetrigine was further studied by doing in silico, in vitro, and in vivo evaluations. So in silico evaluations were done using Tanimoto's structural similarity analysis, which compared the chemical structure of suzetrigine with more than 500 Schedule 1 drugs in the United States with the abuse potential, and it was found that no structural similarity was found between them. Also, the in vitro profiling of suzetrigine with more than 180 non-NAV targets, including GPCRs, ion channels, transporters, and also targets with abuse potential, were found to have no off-target activity at greater than 600-fold margin to estimated clinical concentrations. Suzetrigine was also found to be safe in hERG liability cardiotoxicity studies with an IC50 of 22.9 micromolar, way higher than the positive control terfenadine. Also, when the in vivo studies were performed by doing the CNS evaluations in monkeys and the clinical observations in rats and monkeys, so no stimulant or sedative effects of the suzetrigine exposure were found. So we can say high selectivity of suzetrigine for NaV 1.8 channel and its mechanism of action translated to a well tolerated profile in vivo with evidence of addictive potential, with no evidence of addictive potential. So that was my part. Now I would like to call Stanzin, who will be going to talk about the clinical trial journey of suzetrigine. Thank you so much.
Good afternoon, everyone. So before we begin, I would like to thank my colleagues for setting the stage for suzetrigine by beautifully explaining the chemical concepts, the mechanical aspects, and the preclinical studies which led to the approval of suzetrigine for clinical studies. Now, I would like to take you through the clinical journey of suzetrigine, the trials it went through, and the outcome of the trial which finally led to its approval as a drug. But before we begin, I would like to give a short background about the state of acute pain in America. So Vertex Pharmaceuticals, who were the main stakeholders and were sponsoring the clinical trials for suzetrigine, most of the subjects that they recruited were Americans. So having an idea about the status of acute pain were an important aspect for them. So they reported that there are nearly 80 million acute pain patients every year in the US, out of which nearly 40 million acute pain patients receive an opioid drug every year, because these are the only indicated medications available for severe to moderate acute pain. Now this results in the development of various opioid related disorders, as there are nearly 85,000 individuals every year that develop opioid use disorder, and it becomes a burden to the US economy, as nearly $180 billion estimated annual costs are related to OUDs, out of which $60 billion are attributed to the healthcare for the management of OUDs. So suzetrigine is one such drug that has the potential to address all the concerns that I mentioned. Now, during the clinical journey of suzetrigine was offered with various FDA designations, including the Fast Track designation, Breakthrough Therapy designations, and the Priority Review. This helps suzetrigine accelerate the development, review, and faster approval of the drug as a first-in-class non-opioid analgesic. Now, moving on to the clinical trials for suzetrigine, a total of 3 clinical trials have been completed, and the 4th phase trials is still ongoing. Talking about the phase 1 trial, it was conducted to study the safety, tolerability, and the human pharmacokinetics. And in this trial, a small group of healthy volunteers were taken. In the phase 2 trials, various doses of suzetrigine was given to assess the safety, efficacy, and tolerability. And here, more than 200 to 300 individuals were taken, which were having a pain conditions resulting from the abdominoplasty and bunionectomy surgery. In the phase 2 and in the phase 3 trials, more than 1,000 individuals are taken, and a single dose of suzetrigine was given to again assess the safety, efficacy, and tolerability in again the same 2 pain conditions, abdominoplasty and bunionectomy. Phase 4 trial is a post-marketing surveillance trials to gather the information on the long-term safety and effectiveness of suzetrigine. Now let's discuss each of these trials one by one in detail. In the phase 1 trial, 2 of the basic studies that they conducted were single ascending dose study and the multiple ascending dose studies. These studies were conducted to assess the safe dose range and the human pharmacokinetic parameters, and to know how the body processes and tolerates the drug. So if we talk about the single ascending dose study, a range of single doses were given to assess the Cmax, that is the maximum plasma concentration, and the AUC value, that is the total drug exposure in the body with respect to time. So what they found was that the AUC value was proportional to the dose regimen up to 171 mg, suggesting that the drug follows linear proportionality. And it is an important parameter to predict the plasma drug concentration. However, in the multiple ascending dose study, when the dose was given at multiple times up to 14 days, the Cmax and the AUC value was proportional to the dose regimen up to 65 mg, and beyond that there was no drug proportionality. So they concluded that at the clinical dosing regimen, that is at 100 mg, the drug behaved predictably well in the single ascending dose study, but it was less than dose proportional in the multiple ascending dose study. Now these 2 studies were also important in terms of deciding the dose prescription of suzetrigine, which is the initial dose of 100 mg and followed by 50 mg every 12 hours. Apart from that, they also conducted various other studies like the mass balance study to check the ADME profile of suzetrigine, and it was found that suzetrigine is mainly metabolized by cytochrome P453A enzyme, and the primary metabolite is M6 suzetrigine. Also, they found that there was greater than 94% of the administered dose was recovered, suggesting that suzetrigine has an efficient clearance mechanism. And suzetrigine also has an extensive tissue distribution, and the half-life of suzetrigine was 24 hours, with the time it takes to reach the maximum plasma concentration, that is Tmax value, of 3 hours. They also conducted various drug-drug interaction studies to look how co-administrations of various drugs with suzetrigine is affecting the PK values of suzetrigine or not. And here, in most of the cases, they found that there is no significant effect on the PK values of suzetrigine, but there were some drugs such as strong and moderate CYP3 inducers like rifampin, and strong CYP3A inhibitors like itraconazole, which were significantly affecting the PK values of suzetrigine. So individuals have suggested that they should avoid co-administration of these strong inhibitor and inducer of CYP3A with suzetrigine. Also, in the food effect study, it was found that there was a significant drop in the initial plasma concentration and also delay in the onset of pain relief when suzetrigine was administered with high fat meal and moderate fat meal. So again, individuals were suggested to avoid consuming food from 2 hours before and until 1 hour after the first dose of suzetrigine. So this was all about the phase 1 trial. Now moving on to the phase 2 trials for suzetrigine, a total of 2 parallel studies were conducted. Both were randomized double-blind placebo-controlled dose ranging study. So what does this mean? This means that all the individuals that they taken were randomly distributed in the suzetrigine group and the placebo group, and neither the researcher nor the participant knows who receives a placebo and who receives the real treatment. And also the effect of suzetrigine was compared with that of the placebo, taking it as a control, and a range of doses of suzetrigine was given. The findings of this was published in the New England Journal of Medicine, and their main goal was to evaluate the treatment effect of suzetrigine in 2 pain conditions, that is abdominoplasty and bunionectomy, and they also assessed the safety and tolerability of suzetrigine up to 48 hours of treatment. Now talking about the study design for this trial, in case of bunionectomy, more than 250 individuals were recruited. So bunionectomy is a surgical procedure in which the bunion, which forms at the base of the toe, is surgically removed. This gives a painful condition to the patient. So post-operation 3 doses of suzetrigine was given, high dose of 100 mg, mid dose of 60 mg and low dose of 20 mg. In case of the abdominoplasty trials, more than 300 individuals are taken. So in this case also, abdominoplasty is a surgical procedure to remove the excess fat or skin from the body. So post operation 2 doses were given, 100 mg high dose and 60 mg low dose. In both the trials, an active standard drug was also given, that is hydrocodone bitartrate acetaminophen combination, and for placebo sugar pills were given. And also ibuprofen, which is an established standard pain relief medication, was given to those individuals whose pain becomes unbearable as a rescue medication. Now how did they measure the pain? They measured the pain using a numeric pain rating scale at 19 time points. So this pain scale goes from 0 to 10. So the higher the individual rate their pain, the greater is the pain intensity. So those individuals who rate their pain above 4 was considered to have moderate to severe acute pain. Now talking about the endpoints, their primary endpoint was to determine how people's pain decreased over 48 hours of treatment in suzetrigine as compared to placebo, that is the SPID-48 value. So SPID-48 is a numerical value which indicates the extent to which the pain is reduced. So for our understanding, we can say that higher the SPID-48 value, greater is the reduction in the pain. This table shows the average SPID-48 value of all the individuals in both the trial. And here we can see that at the high dose group there is a higher value of SPID-48, suggesting that at high dose there is a greater reduction in the pain. Similarly, their secondary endpoint was to assess how people's pain decrease after 24 hours, that is SPID-24 value, and in this case also at high dose group there was a greater reduction in the pain in both the trials. So suzetrigine at the high dose group reduced acute pain more effectively over a period of 48 hours after abdominoplasty and bunionectomy. Now talking about the phase 3 trials, a total of 3 trials were conducted, out of which 2 were parallel studies. Both were randomized, double-blind, placebo and active control drug. This time, apart from comparing the effect of suzetrigine with placebo, they also compared the effect of suzetrigine with the active drug, that is hydrocodone acetaminophen combination. And the findings were again published in the journal Anesthesiology. Study design was almost similar to that of the phase 1 trial. However, this time more than 1,000 individuals were recruited and a single dose of suzetrigine was given, which was found out to be effective in the phase 2 trial. Again, as a active standard control drug, acetaminophen hydrocodone combination was given and ibuprofen was given as a rescue medication, and pain was again measured with the help of NPRS rating scale. So their primary endpoint was again to compare suzetrigine with that of placebo by using the SPID-48 value. However, this time, apart from considering the effect of suzetrigine alone, they also considered the effect of ibuprofen, which was given as a rescue medication. So in both the cases, there was a significant reduction in the pain as compared to placebo, and this is indicated by the positive value of SPID-48 value. So suzetrigine had a greater reduction in moderate to severe acute pain as compared to those taking placebo, and it is similar results are also obtained when the effect of ibuprofen rescue is also considered. Next, they also compared the effect of suzetrigine with the active control drug, that is hydrocodone acetaminophen combination. Now, this is a type of non-inferiority trial which aims to show that the new drug treatment, that is suzetrigine, is as effective as the standard drug. So here again they found that suzetrigine performed almost similar to that of the standard drug in terms of effectiveness. However, individual would prefer suzetrigine more because of its non-opioid nature, due to which it led to less side effect and high safety margin. Lastly, they also checked the time it takes to get a clinically meaningful pain relief, that is the time it takes to get greater than 2-point reduction on the NPRS scale. And here again, when the effect of suzetrigine was considered alone, there was a significant reduction in the time as compared to placebo. However, when both ibuprofen and suzetrigine was compared, the time even further got reduced. So subjects who were receiving suzetrigine achieved clinically meaningful pain relief rapidly in participants receiving suzetrigine as compared to placebo, and it was more rapid in case of multimodal therapy. That means ibuprofen effect was also considered. Later, a separate single-arm study was also conducted in various surgical and non-surgical patients. So single-arm study means that all the individuals that were recruited were giving the same treatment of 100 mg suzetrigine, and this treatment went on for 14 days. So their main outcomes were to assess the safety and tolerability and to check the proportion of participants who were rating suzetrigine as good or excellent. So in terms of safety, suzetrigine was generally safe and well tolerated, and most of the events were non-serious and easily manageable. In terms of effectiveness, 82% of the surgical patients and 91.2% of the non-surgical participants rated suzetrigine as good or excellent. So this study provides a supportive data regarding the safety profile of suzetrigine, especially for longer term use given the period up to 14 days. Now, after all the clinical studies, there were some adverse events, and if we look at the safety and side effects, however, there is no serious side effect or any death that was observed that could lead to discontinuation of the study, but there were few common side effects, such as in trial 1. Most common side effects were depressive mood, dizziness and somnolence, and in case of trial 2 and 3, most common side effects were itching, muscle spasm, rash and dizziness. There were also incidents of nausea and vomiting in case of the abdominoplasty and the bunionectomy trial. However, if we look at the percentage, it is lower in case of suzetrigine as compared to placebo and the active control drug. With all this, now we can safely say that for the first time in over 2 decades, suzetrigine, a novel small molecule and a highly selective NaV1.8 inhibitor, offers an effective and a safe non-opioid, non-addictive treatment with broad applicability for the treatment of moderate to severe acute pain. With this, I would like to end my talk by showing a short video clip to know what clinicians think about suzetrigine. Thank you.
The FDA approved the first medication for pain as a non-opioid medication in the last 20 years. This medication is called suzetrigine. It's a sodium channel blocker, one of the most important channels of the nerve cells. Without it, the nerve cells cannot conduct pain signal. 18 million people suffer from acute pain, whether from surgery, trauma, or other injuries. The current mainstream treatment is opioids. From a scientific perspective, I believe this is not going to be an addictive medication because it's a sodium channel blocker. This has been used, it's not the only one. Some other medications, including the anti-seizure medication, for example. One of the most common mechanisms for anti-seizure medications, sodium channel blockers. So therefore, those medications do not have addiction property. So this one, I do not see. The FDA.
With this, I would like to conclude this part of the session. Thank you.
Thank
Thank you, Team Suzitrizine, for this wonderful presentation. We hope the symposium was not only informative but also inspiring, a reminder of why we pursue science and what we can achieve through collective efforts. With that, we formally conclude the student presentation segment of this symposium. Now, it's my honor and privilege to invite our chief guest, Dr. Geetha Vani Rayasam. She played a pivotal role in shaping science based policy frameworks and translating research into real world impact, particularly in areas related to societal development and public health. Her visionary leadership, multidisciplinary approach, and commitment to inclusive science have been deeply inspiring to the scientific community. Ma'am, we would request you to please share your thoughts. Over to you, ma'am.
Yeah. Good afternoon, everyone. I hope I'm audible.
Yes, ma'am, you are.
So, firstly, congratulations to CDRI, to all the students and the director for such a wonderful session. I really enjoyed the whole session today, and I think it's really nice that this is a symposium for the students and by the students. And I think this is one of the unique things I have attended in the recent times. It reminded me of my PhD days, where we used to have journal presentations, where we used to talk about the research papers where the students would present. But what you students have done today is go beyond the papers, but actually talk about the entire drug development, which I think is very relevant, not only to CDRI, but to all of us. Like one of the students pointed out, a small pill, which we take it so easily, the painstaking efforts which go beyond the discovery and the development. Not many people understand what it takes to develop a drug. And today we are blessed with so many options, and that longevity has increased. And this is a miracle of the science, if I can say. And it has been very well highlighted by these two case studies presented by the students. And I really compliment the students for such wonderful presentations, but full of confidence and poise. And I think the future is bright for the country. And I really enjoyed this. And I must say that recently I also heard some of the school students during our Jigyasa Epic Hackathon. They also presented very well. So across the spectrum, I see the youth of our country really rising up to the occasion. And I think science has a very good future in our country, with all of you taking up such interesting research problems and talking about it. And I think one of the things which was really highlighted is how many decades it really takes to develop the drugs, right? And even today's presentation, it was a team effort. Like, each of you talked about one area. And even in real life, when you try to do drug discovery, no one person can do the entire drug discovery. And I think that's a thing which is embodied even in CDRI or in any drug discovery industry. It's a whole team effort. And I think that's one of the learnings for each and every one of us, that teamwork, interdisciplinarity, you need to know chemistry. Like, it's not that everybody can do everything, but how multidisciplinary it is, from pharmacokinetics, pharmacodynamics, clinicians, to the pharmacologist, to the biochemist, molecular biologist, computational work. So the expertise required is very varied and vast. And I think no single person can achieve it. And I think it's this teamwork which is critical, and the handholding. And I think this is one of the lessons both for the students and for all the scientists as well, that how much of teamwork is required to make a successful drug. And many times, many of them are unsung heroes. And I think today we told two successful stories. And as we know, drug discovery is not an easy process. For every 2 molecules which make it, perhaps thousands of molecules never make it. But there is a lot to be learned from that as well. And so one of the things and another request to the students would be, also develop critical thinking. Like, when you're presenting this, did you see any shortcomings in the study? So one is to, okay, these are FDA approved. That's really nice. But we should also look at critically, what could have been done differently. I think that's something which the student should also develop in terms of questioning and, looking at critical data analysis. That is something which will be really useful going forward. And another request to the students, I think you people spoke very fast. I think especially if there are students or audience who is not from the area, for them it would be, a bit challenging to understand. I think a little bit slower, and I think all of us Indians, we face this problem. We all speak very fast. So I think this is just a suggestion from my end that especially for audience who is not specialized. And these were two comments which I wanted to talk about. And so again, I come back to the learning. So many failed clinical trials also teach us a lot, many of the failed drugs. So it's also important to, keep those in mind, especially when you're designing or doing your drug discovery. And it is especially challenging. I can understand, in academic setting, doing drug discovery. And having said that, CDRI, I think over the decades has really been doing a tremendous job in terms of drug discovery and development. It's a unique organization which is has end to end. And I think there are many success stories. And it was very interesting to hear about Picroleaf. And I really do hope that trial, phase 3 trial is successful and gets approved. And we should benchmark globally. And I think our ambition should be for globally benchmark drugs, and which I'm sure CDRI will be able to achieve in years to come. And Picroleaf could be one of the leading examples. And and I think many times we have this, many people don't want to do applied science or translational science and basic science. And there's always debate, what is interesting. But I think there is no such thing as a, applied science or a basic science. It's good science versus bad science. And if you see, many of these papers have come in leading journals. So it's not as though that you cannot publish if it is a translational science or an applied science in a good journal. I think it's a good science which matters, and I'm sure you all will be able to not only do the translational component but also achieve academic, laurels, right from your PhD to various careers. And coming to careers, again, I think one point which I would like to highlight to the students is not only academic careers, there are many other careers open. Maybe many of you may become entrepreneurs of your own, have your own startups. And, I could see that some of you could become CEOs of a company tomorrow. So the path is wide and vast, and especially with this kind of exposure and research experience, I'm sure, apart from academics, industry, and many other sectors are open to all of you. And I would like to finally conclude that, so as I was told, and it is very nice to see the JRFs and SRFs. And many times we are asked, what is this fellowships you are doing? What is the impact? And I think this is the impact where we are training young scientists in the country. And if you go, like I was in industry, and every time, CSIR people are there, especially from CDRI. I have seen, in drug discovery, there were PhDs from CDRI who are working in industry. And one of the contributions of CSIR has been, probably there's not a single organization in the country which is dealing with science and technology with somebody from CSIR associated is there. And I think this is the single most contribution of CSIR apart from its technologies. And you may be happy to know that CSIR has supported more than 1 lakh students from the beginning over the years through fellowships, through various projects. And as you know, and we are here to troubleshoot. I understand that there were problems in fellowship disbursement, especially during COVID and post-COVID. But to a large extent, we have kind of contained them. It can never be zero because there are thousands of students. And if many of your students who are CSIR fellows are listening, one request is, I know much of it is bureaucratic, but please follow the processes because it's important to submit your reports on time so that you get your fellowship on time. Many times, I know you're busy with the experiment and you'll think this is not important, I can do it later. But please follow the guidelines because that will help us also to disburse the fellowship on time so that you don't feel the pain. So I know, although it's a boring job, but please make sure that you follow the deadlines, and also request to the scientists and the PIs that please make sure that the students submit the reports on time. And so that, there's a smooth experience for the students while doing the research. And so these are my few of my comments. And again, I'm sorry I could not be in person because of my urgent engagements. And again, I would like to thank Dr. Radha and Dr. Amir for giving me this opportunity to be here and to listen to the excellent presentations by the students. And I wish you all the best. Thank you.
Thank you so much, ma'am, for your insightful words. Your ideas and encouragement have added great value to this event and have certainly inspired our young scientific community. Now, I request Director CSIR-CDRI to please come up on the stage and share your thoughts about the event. Director, ma'am, please.
I want to congratulate all the students who presented today. The storytelling was exquisite. The synthesis was, beyond expectations. And the learning experience for us sitting in the audience was fantastic. So one more round of applause. They're all here. Okay, I see you. Okay, wonderful. I really commend you on all of the hard work that went into it. And one thing that was amazing is that we said that 50 minutes and no more. I said 45 minutes was okay, but 55 minutes was not okay. And you really ran it like clockwork and finished at 50 minutes. Because you were able to do that, I know that your practice and your rehearsals were great. And this is the other point, that communicating, and for that matter, anything in science, anything in life, is about doing things again and again and again till you become so good at it. Still, you have that expertise, the confidence, and the ability to tell the story, and you can do it within the time that you have. On many counts, this has been a fabulous experience. I hope we can repeat this every year. I know that it took a lot of effort on the part of the mentors, so I do want to specially thank them. They put in very serious effort. As you know, this is the first time we're doing it, and the reason for doing it is something like this. For students who are getting their PhDs at CDRI, it is very important that you all learn, whether it is firsthand or it is by watching, what exactly it takes to develop a drug. Because when you go out into the world and you carry a PhD from the Central Drug Research Institute, you need to be able to stand up there and say, yes, I can tell you something about drug discovery and development. And you can only say that if either you are part of a drug discovery project, which sometimes I know is difficult as students, because you have other things to also think about in terms, for example, publications and so on and so forth. But to be involved in some manner, maybe by watching, maybe by attending team meetings, maybe by looking at the progression of compounds that perhaps your PI is involved in. This is important for you. And the other way of learning is by seeing how somebody else developed drugs. And that learning is very essential because if you think about how difficult it is to do drug discovery, I can confidently say that no textbook will teach you this effectively. Right? And you can learn about the theory, but the nuances, the application of a particular concept or a hypothesis or an assay really depends on that particular project you're working on. It depends on the target. It depends on the indication you're trying to address. So one learns really by doing. And in your lifetime, you may not be able to work on many, many, many drugs. But you have the opportunity to learn by reading about other drugs and apply that back into what you do. So this was the beginning of that process. I'm really, really thrilled and delighted that you all students who participated and all of you in the audience did so with so much enthusiasm. And therefore, it gives me confidence that, we can keep doing this perhaps once every year. So thank you so much. Thank you to the audience. Thank you to those online. Thank you, Geetha. We really appreciate the fact that you took time out in large part of your afternoon. It means a lot to us. We see how committed you are to the learning experience of students, to lifting the level of science, to promoting R&D in India. This is our small effort in that direction, and it matters so much to us that you joined us and supported this endeavor. So I would now like to move to the final part of this this event today, and that is felicitations. Amir, are you here? So let's bring our speakers back on stage and felicitate them. Amir?
Yeah, please stay on stage.
Yeah.
What an awesome beginning to this wonderful series that we started during this Platinum Jubilee celebration, and will continue once every year. Thank you, ma'am, for guiding us into this, and we thank our chief guest, Dr. Geetha Vani Rayasam, for spending her afternoon with us. So we were expecting you here, ma'am, and we had some small token of appreciation for you, which I believe whenever you happen to visit us next, we will pass it on that time. So moving on, it's time to felicitate the stars of the day. And I would like to invite all the students. Hope you're around. Okay. So first up in this sequence, please come up, Miss Swecha from Team Resmiteron. And I would like to request Director Ma'am to do the honors and present a small token of appreciation to the students. Astha is next. Followed by Miss Astha, please. Miss Priyanka, please. Miss Neha. From Team CSIR-HRDG, we have Miss Adra Chakraborty. Miss Lovely Dikshit. Mr. Ashutosh Sharma. Yeah. And Mr. Stanzin Nurgu. Okay, sir. Yeah. The most of the mentors did not want to come up on stage, but ma'am insisted, and she has a small token of appreciation. So, yes. So, Dr. Anil Gaikwad, Dr. Kumara Velu, Dr. Ajay Kumar Srivastava, Dr. P.N. Yadav, Dr. P.P. Yadav, and I also would not like to miss this opportunity. So, I will be joining as well.
Call upon you, Dr. Amir Nazir.
I'm already here.
You guys are the stars behind the scenes. Thank you.
Go ahead. Anil first. Kumar. Ajay. PN. PP. PN. PP. And there is one. So we'll have a group picture. Yeah. So please, all the students, we'll have a group picture for all the participating students, mentors, along with Director Ma'am. I will have it as well.
Yes, ready. Thank you.
Thank you. May I now invite Ms. Priyanka to please propose a vote of thanks. And with that, we'll conclude the program. Please be seated, audience.
Thank you, everyone. So, I think this symposium was designed as a platform for young researchers to explore and present their understanding of FDA-approved drugs. And what we witnessed today was a true reflection of that goal. I would like to extend our heartfelt gratitude to our chief guest, Dr. Geetha Vani Rayasam, for graciously sparing her valuable time to be with us today. A sincere thank you to our director, CSIR-CDRI, for her constant encouragement and for nurturing an environment where scientific curiosity and student-led initiatives are truly valued. Ma'am, thank you so much. Your support continue to inspire and empower us all. Behind every confident student, we saw today stood a mentor who patiently guided them and helped shape their scientific voice. Thank you to all the mentors present here. I'd also like to thank Mr. Ramindranth and Mr. Kazim, who were always there to beautifully capture each moment of the symposium. A big thanks goes to the backstage team and student volunteers Aniket and Maria, who worked tirelessly behind the scene to ensure everything flowed smoothly. And finally, I would also like to take a moment to acknowledge and thank our distinguished guests from various academic institutes who have joined us today online and offline. Your participation has added great value to this event, and we are truly grateful for your enthusiastic presence and support. Once again, thank you all of you, especially our in-house faculty and students of CSIR-CDRI for being a part of today's symposium. With that, we conclude the event. Have a wonderful evening ahead. Thank you, everyone.